So welcome back to the meiosis process. We are going to talk about amazing process, which is meiosis. So meiosis actually is the first division in the process of the making gametes. So what are the gametes? Gametes, gametes, they are sex cells. They are sex cells. The second name of the gamete sex cell. It's divided into two. The first is sperm cell. The second is ovum egg cell in the female, sperm in the males. Okay, so this is meiosis one. Actually, the meiosis divided into two phases: meiosis one and meiosis two. So meiosis one is the first division in the process of making the gametes, sperm and egg cells. It reduces its main function. Main function of the meiosis is to reduce the chromosome number from diploid to haploid. If the diploid number of human body is 46, the haploid number will be 23. Okay, so meiosis main function is to reduce the chromosome number from 46 to 23, from diploid number to haploid number. Meiosis 1 includes 4 phases. They are prophase 1, metaphase 1, anaphase 1, telophase 1. Okay? So before watching the meiosis process, I suggest you to watch the mitosis, because meiosis 1 is actually similar to the mitosis. So cellular processes, the stages of each process is similar to the mitosis, but there are some additional ones in the prophase 1. So the biggest difference in meiosis 1 and meiosis 2, actually, the biggest difference between them: in meiosis 1, the homologous chromosomes, they pair up. Okay, this never happens in mitosis. But in meiosis 1, the homologous chromosomes, they're going to pair up. Meiosis divided into two phases: prophase 1, metaphase 1, anaphase 1, and telophase 1. Okay, this is the biggest difference. So what's happening in the prophase 1? Let's talk about the prophase 1. So in the prophase 1, it's not just a simple version of the mitosis prophase. So they are the similar, for example, chromosome condensation, disappearing of the nuclear envelope, and also disappearing of the nucleoli, formation of the spindle fibers, migrating of the centrosomes to the poles. They are exactly the same with the mitosis in the meiosis 1 as well, in the prophase 1 specifically. But there are some additional things. So 4 additional things. First, 3 additional things: synapses, tetrad formation, and the crossing over, pairing up of homologous chromosomes. They are additional 4 main things in the prophase 1, okay, which is happening. So actually, the synapses is happening. First of all, what's synapses actually? So synapses actually, as you can see, first of all, happening the homologous pair. So each pair of the chromosome, one set is came from the dad, one is came from the mom. They're going to pair up. So homologous chromosome means they are different in colors here. They are but they are the same in their shape. They are same in their size. They carry the same genes of the chromosomes. You see, they are in same length, same shape, and also same size, and they carry the same genes, but their main difference they're in their alleles. For example, allele is a part of the chromosome or DNA, which is responsible for any physiological appearance. For example, this part is responsible for the brown eyes, and this came from, let's say, from the mom, and this part is responsible for the let's say blue eye color, blue eyes. Yes, let's say blue eyes. So this came from the dad. They are homologous. They have the same gene in the same locus, in the same location. They have this in the same location, same genes, but the difference in their gene content. We say it alleles. Okay, they are different in their alleles of these genes. So these are homologous chromosomes, 2 chromosomes which are identical in their size, in their shape, in their genes, but different in their alleles. Okay, this is called homologous chromosomes. So homologous chromosome pairing up, this happens in the prophase 1. So this pairing up of the chromosomes and coming up closer, so is called synapses. Synapses is homologous chromosomes find their match and they are pairing up. This is called the synapses. Each human cell has 23 pairs of the chromosomes, right? During the synapses, all those 23 pairs, they're going to find their partners, forming 23 homologous pairs. Okay, this process takes a long time. So prophase 1 is the longest phase. Okay, so you can see synapses, they are pairing up. Homologous pairs are coming close to each other. They are pairing up. This is called what? This is called synapses. So why is this happening? Without the proper synapses, chromosomes won't line up or exchange genes correctly. They should exchange the genes, but without the synapses, they cannot do it. Okay, chromosomes won't line up together. This leads to the errors like infertility or other genetic disorders. That's why synapses is crucial. So synapses is a main difference from the mitosis. Okay, so what's crossing over? Crossing over, this is exchange of the genetic material between non-sister chromatids of homologous chromosomes. So what does it mean? Exchanging of material between two non-sister chromatids. So this is one sister chromatid, this is second sister chromatid, third, and the fourth sister chromatid, right? So they are homologous chromosomes. This is homologous chromosome, but exchanging of non-sister chromatids. This sister chromatid and this sister chromatids, they are called non-sister chromatids, right? They are not sister chromatids to each other. They're going to come together in the process called synapses, and they're going to exchange their genetic information in deadlocks. As you can see, this chromatid and these chromatids, they are exchanging their information between two homologous chromosomes. And this exchanging of the genetic information is called crossing over. So crossing over, this is exchange of the genetic material between non-sister chromatids of the homologous chromosomes. And this is always happening. This is important. This is always happening after the synapses. Okay, this results in the new gene combinations. Okay, so now they will have new gene combinations in each chromosome. They will have new gene combinations because they exchange it. Mom's chromosome and dad's chromosome, they exchanged their information. Now they have new gene combinations that are genetically different from both parents. They are not genetically same to parents. They exchanged their genes, and that's why they are now genetically different from both parents. Okay, and this because this crossing over makes each sperm and egg cell unique. Each of your sperm cell or each of your egg cell or ovum, they are unique. All of them are unrepeatable. That's why you're unique. You don't look like to other people. 7 billion people in our planet, 7 million of them are different. Okay, all of them different because of the crossing over. Okay, that's the case. So you have to know. So what's tetrad formation? So let's talk about the tetrad of the chromatids. So what's the meaning tetra? Tetra means four. Okay, each homologous pair has four chromatids, right? As you can see, this is the homologous pair. Each of them have four chromatids. This is one chromatid, second chromatid, third, and fourth. These four chromatid, each homologous pair has four chromatids total, two chromatids in per one chromosome, two chromatids per one chromosome. So two chromosomes will have four chromatids. This four-part structure is called tetrad. Okay, you see, this is one chromatid, second chromatid, and this is homologous pairing already happened. And you see homologous chromosome behind the bluish color. This is the third chromatid, and this is the fourth chromatid. So four chromatids together, they are forming the tetrad. Tetrad is formed here. Okay, so tetrad means four chromatids together. So tetrad formation is also happening in the prophase. I hope that was understandable. So medical relevance. What's the medical relevance? Defective synapses means it leads to the infertility if homologous together they fail to pair up. If they fail up to pair up, gametes may have abnormal chromosomes. Okay, and the crossing over errors leads to the chromosomal disorders. Improper recombination that causes translocations and the duplications. So That's why it's really important to know. Okay, so we talked about the synapses, tetrad formation, crossing over, and chromosomes are condensing in the prophase one, and spindle fibers are forming, nuclear envelope is disappearing. You see, nuclear envelope is disappearing here, and spindle fibers are formed, and they are going to the poles, the centrosomes going to the poles, and the chromosomes are condensed. They are visible under the microscope now. Okay, this is you should know about them. So please answer to the comment section. What do you think? Which event makes the prophase one the longest phase? If you know it, please write to the comment section. And my second question is, how is prophase one differ from the prophase of mitosis? In the comment section, please write down the answer if you know. So this is the prophase one. You see the maternal chromosome, paternal chromosome, they're doing the synapses and crossing over. Now you have the recombinant chromosome. We call it recombinant chromosome because they exchanged their genes. I hope you understood. So metaphase one. So we know the function of the meiosis one. This is to reduce the chromosome number from diploid to haploid by separating the homologous chromosomes, not the sister chromatids. Don't confuse. Our main task is to, in the prophase one, we did what? We did actually the pairing up of homologous chromosome. But then our task in the meiosis one actually to separate those homologous chromosomes to get two cells which have half number of the chromosomes. Okay. So in the metaphase one, what's happening in the metaphase one? The 23 homologous pairs. Let's say here we have two pairs of the homologous chromosomes, right? They are lining up. Those tetrads are lining up in the metaphase plate or equatorial plate. We talked about the metaphase of mitosis. Please watch if you haven't watched it. Now, not individual chromosomes lining up in the metaphase plate. Do you remember in the mitosis, individual chromosomes line up in the metaphase plate? But in the meiosis, the homologous pairs, they pair together, the homologous chromosomes, right? The homologous pairs are going to line up. Okay, this is the metaphase of mitosis. This is the mitosis metaphase. This is the metaphase one of meiosis. You have to know, you have to differentiate between them. Okay, so in the human body, 23 pairs, homologous pairs, we call them tetrads, lining up along the center of the cell, which is called metaphase plate or equatorial plate. Okay, they are lined up in pairs, not individually like mitosis. Okay, two X-shaped chromosomes side by side is illustrated in the diagram here. Okay, you have to know about it. So pairs are located in the center. As you can see, this is the metaphase. This is the metaphase of meiosis. This is the metaphase one of meiosis one. This is the metaphase of mitosis. Individual chromosomes are lining up here, but here the homologous chromosomes are lining up here, and spindle fibers are attaching to each chromosome centromere, okay? And the centrioles are already in the poles here. And this all was about the metaphase. This is the illustration of the metaphase. As you can see, each spindle fiber is attaching to one homologous chromosome. So, but we don't know which chromosome came from that, which one came from mom. So spindle fibers can attach to this one, or this one can attach to this one. We don't know to which pole those chromosomes are going, or to which chromosome those spindle fibers are attaching. We don't know. That's why this is called independent assortment. Okay, independent assortment. So those spindle fibers, we don't know to which chromosome they attach and which chromosome goes to which pole. We don't know about it. This is also gives you genetically difference from your parent. Okay, this is the second reason. The first reason was crossing over. The second reason will be we don't know independent assortment. We don't know which chromosome goes where. Dad's or mom's to the left or right, we don't know to which part it goes. This is the second reason why we are genetically different from our parents. Okay, the main difference now 46 chromosomes line up in the center. So 46 individual chromosomes line up in the center. This is in mitosis, in the metaphase of mitosis. 23 pairs of the homologous chromosomes lining up in the center. This is happening in the meiosis I. What's happening in the anaphase I? Anaphase I, what's happening here? Centromeres not splitting here. Centromeres are still there. You see the centromere in the center, they are still with chromosomes. Okay, they are still attaching the sister chromatids on their places. Okay, but the main differences of anaphase, instead, each homologous chromosome is pulled to the opposite poles. Okay, with their two chromatids. Each homologous chromosome, we are dividing the homologous chromosome from each other. We are disturbing the synapses now. Okay, so each homologous chromosome is separating to each pole. So each side gets one full chromosome from each pair. Okay, so this chromosome and these chromosomes, the centromeres are still there, sister chromatids are still there, recombinant chromosomes, they are now detached from each other. Okay, so you don't have to confuse between the anaphase of mitosis when the sister chromatids separate. This is the anaphase I of meiosis. So you shouldn't confuse between them. Okay, as you can see, recombinant chromosomes, all of them, after what happened here, they are recombinant chromosomes, they did the crossing over, now they are separating from each other. Homologous chromosomes are separating from each other, but still sister chromatids are still there. So what's happening in the telophase I? In the telophase, spindle fibers are going to disappear, nuclear membrane again reforms, right? Nuclear membrane is reforming, the chromosomes are there with centromeres with themselves. Chromosomes start to uncoil, you see they are uncoiling, unraveling, or we can say, okay, and cytokinesis is starting here if it's animal cell or human cell. Cleavage furrow is forming. If you don't know cleavage furrow, please watch the previous video I have explained. But each cell still has duplicated chromosome. You see, each cell, when we get individual cells, when we get at the end individual cells, so here the cleavage furrow formation is happening. So each cell now has duplicated chromosomes. Sister chromatids are still together. Each new cell is now haploid. Before that, in one cell, we had 4 chromosomes. Now, if we divide this cell into 2, each cell will have 2 chromosomes respectively. So this is the haploid in number. We call it N, okay, 2. But here we had N is equal to 4, okay? So you shouldn't confuse between them. So the cells are not identical to each other because of the crossing over now, and each cell has now in this case 2 chromosomes, but in human cells it will be 23 chromosomes each cell, made of 2 chromosomes. Okay, even though they still look like X, they are haploid, only one set of the chromosome. Okay, so the key events which are happening in the prophase I, synapses and the crossing over is happening. Okay, so in the metaphase I, homologous pairs align in the center. In the anaphase I, homologous are pulled apart to each poles. In the telophase I, cells split into 2. Okay, so cytoplasm is going to divide, cytokinesis is happening. Okay, if you have any questions, you can approach in the comment section. So this is telophase. You see sister chromatids are still there. I try to illustrate it in 3D structure. Okay. So this is all about the meiosis I. So this is the diagram which explaining you the difference. At the end, you will have 4. At the end of the meiosis I, you will have 2 cells haploid in number. In the cell before we had 6 chromosomes. Now each cell is having 3 chromosomes respectively. Okay. So you can read this chart by yourself. I will share the slide. So meiosis II, let's talk about the meiosis II. Meiosis II is like mitosis, but it happens in haploid cells. Meiosis II is looking like the mitosis, but mitosis is happening in diploid cells. But in the meiosis II, it's happening in haploid cells. You start with 2 haploid cells. Okay, you're starting with two haploid cells. This is haploid in number. This is also haploid in number. Two haploid cells. You're starting meiosis II, each with 23 chromosomes. Imagine this is human cell, each having 23 chromosomes, but each chromosome still has two sister chromatids. Each of them has two sister chromatids. Okay, meiosis II separates those two sister chromatids. Okay, as you can see, they are separating those two sister chromatids in the anaphase II. As you can see, these are your sperm or egg cells, which are genetically unique. At the end of the meiosis II, you're going to get four haploid cells. Okay, so meiosis II phases: prophase II, metaphase II, anaphase II, telophase II, and at the end, cytokinesis. Okay, so what's happening in the prophase II? In the prophase II, so spindle fibers, they form again, nuclear envelope break down. Everything we have learned in the mitosis is happening here. Chromosomes prepare for another division. They are already X-shaped from the meiosis I. Okay, no synapses, no crossing over is happening in the prophase II. Prophase II is exactly the same with prophase of mitosis. Okay, there are no homologous pairs. Okay, there are no homologous pairs in the prophase II. In the metaphase II, please watch the video of the metaphase of mitosis. In the metaphase II, what happens? Chromosomes, which are 23 chromosomes per cell, they're going to line up singly along the metaphase plate. This is the prophase II. As you can see, there are no homologous pairs. As you can see here, there is no synapses, no crossing over. It's happening. It's already happened. Okay, spindle fibers are going to form and centrioles are going to the poles in this case, and nuclear membrane is disintegrating and disappearing. In the metaphase, you can see chromosomes, they line it up slightly along the metaphase plate. This is the metaphase plate. The chromosomes line it up in the metaphase plate, okay, in the middle. This is like mitotic metaphase, it looks like. Okay, single line of X-shaped chromosomes, one per single spindle fiber, they are attached. So what's happening in the anaphase II? Chromosomes, so here centromeres finally splitting, you see? The centromeres, which were attaching two sister chromatids together, they now splitted. So sister chromatids separating and moving to the opposite poles now. Sister chromatids are moving to the opposite poles, okay? So two cells, of course, not one cell. We have two cells because at the end of the meiosis I, we got two cells. So the chromatids are not identical because of the earlier crossing over. Okay, earlier they did the crossing over. That's why these sister chromatids, they are not identical to each other anymore. So what's happening in the telophase II? This is when the nuclear membrane is reforming. You see the nuclear membrane, it's reforming now, and the chromosomes begin to uncoil. Chromosomes are uncoiling as usual, and the spindles, they disappear. You don't see the spindle fibers here. Now you have two nuclei in each cell, and they are haploid in number. Okay, they are haploid in number. So cytokinesis finally is happening after the telophase II. So cytoplasm divides in each of the two cells from the meiosis I. You now have four haploid cells. You see four haploid cells you're getting at the end, each with 23 unreplicated chromosomes. But here, before we had four, but now you have two. And each chromosome now has only one chromatid. We call them one chromatid chromosome, okay? And they are genetically unique because they did what? They did the crossing over before. And four of the cells which were from the meiosis, they're genetically unique, okay? So this is the best diagram. So DNA synthesis is happening in the S phase of the interphase in meiosis, in the mitosis as well, in the S phase of interphase, okay? Synapses of homologous chromosome is only happening in the meiosis during the prophase. It's not happening in the mitosis. Crossing over only during the prophase one of meiosis. It's not happening in mitosis. Homologous chromosome lining up at the metaphase plate only during the metaphase one, not in the mitosis. Sister chromatids line up at the metaphase plate. It's only during the metaphase two, of meiosis and during the metaphase of mitosis, okay? So number of genetic composition of the daughter cells, 4 haploid cells at the end of the meiosis II, 2 diploid cells at the end of the mitosis. If you know these concepts, if you know this table very well, you won't have any mistakes during the examination. So at the end of the meiosis, you are getting 4 haploid number cells. At the end of the mitosis, you're getting 2, okay? So this is the similarities and the difference of the mitosis and the meiosis. This diagram illustrates the similarities. You can read it yourself. Please read it. So these are the similarities, and this is the difference between the mitosis and meiosis. I hope you will understand by reading. I have explained to you already everything. So meiosis is 2 cell division, but the mitosis is 1 cell division. Mitosis results in 2 diploid daughter cells, but meiosis results in haploid daughter cells. And in mitosis, daughter cells are genetically identical to the parent, but daughter cells are genetically different in the meiosis. And mitosis creates all body cells, which we call the somatic cells, but the meiosis creates only germ cells, which are sperm and egg cell, okay? There is no recombination or crossing over in the prophase, but there is a recombination and a crossing over of the chromosomes in the prophase 1 of meiosis. In metaphase, individual chromosomes line up, but in the metaphase 1, what's happening? Pairs of the chromosomes line up. The pairs, it's important, pairs, okay? Here, individual. During the anaphase, here, sister chromatids are separated to the opposite pole. During the anaphase 1, the sister chromatids move together to the same pole. Sister chromatids are still attached to the centromeres. But during the anaphase 2, yes, sister chromatids are separated to the poles, okay? So this is the quiz. You can do the quiz on your own. Here I have showed you the quiz. I hope it was understandable. If you have any questions, please write down them to the comment section. Thank you very much.